Hello guys, welcome to my YouTube channel. Let's start it together. Today we'll be focusing on the DNA code of life. So we'll start on looking at the structures of DNA and RNA. Actually, it's the differences of DNA and RNA. So these are the types. Okay, let me show you. They say DNA has 4 different nitrogens bases. It's adenine, thymine, guanine, and cytosine, of which when it comes to RNA, though that's type of structures, like that's adenine pairs with thymine and guanine pairs with cytosine. So on the RNA structure, you'll find that um, adenine doesn't pair with thymine. It changes to pair with uracil. So let's continue looking here, guys. So as you can see from the start there, it says adenine is with thymine. Okay, as you can see, it is written down there that adenine always pairs with thymine. Nothing will change on a DNA. Nothing will ever change. So also guanine pairs with cytosine. Always, nothing will change. So you have to master all these in this table so that you'll be able to differentiate between these two, DNA and RNA. As you can see there, that adenine always pairs with uracil on the RNA. So guanine doesn't change on both sides. You should know that. Or what changes, it's the adenine who it pairs with. See? Yes. I think that's clear enough for you guys because it's very easy to look at the structure or to differentiate in this table. So just in case guys, I don't know. So they say below shows the structure of DNA and RNA. Okay, these are words compare and contrast. So we look at DNA and RNA. So the actually of what they're comparing here, the what nitrogenous bases look alike, you know, to them. So here it is. Of what it is present to this boat, we all know adenine, cytosine, and guanine, not thymine and uracil, because they differ from one another. You know what I mean when I say one another, because on DNA you find that there's thymine, and also on the RNA you find that there's always uracil. Doesn't change. You see. So of what is similar to this is just adenine, cytosine, guanine. And you know the difference is, guys, as I told you that a adenine on the what's on the DNA always pairs with thymine. So on the on the RNA, it's always pairs with uracil. So also what's sugar, okay? Don't be confused, okay, on the parts, because some students were so confused about that. On the DNA, guys, the sugar is deoxyribose, and on the RNA, it is ribose. Okay, just be careful with those. Okay, so this, if we're looking at the structure of the DNA, we'll find that it is double helix structure, or you can say it's a double-stranded structure. On the RNA, It is what's a single structure. Okay, be careful with those. Um, Now you can see we've been given here. I can, like, if you want to screenshot this and write and tell and just maybe on the comments below tell me that you have written so that we can correct each other. Good, yeah, man. That's how it is. You see, look at the structure on the DNA. You see why it is a double stranded. Okay, it's double stranded structure. So on the RNA, it's a single stranded. That's simple. Simple, straightforward. Okay, so let's go. Uh, yeah, yeah, I've already told you the differences between DNA and RNA, guys. I did. So we'll now hear on the DNA replication. First of all, I know that you would be like asking yourself of what is DNA replication. DNA replication takes place where? On interphase. Don't ever go to the paper without knowing that. Okay, they might ask you, maybe it's just a 1 mark question, sometimes 2 marks, but you have to know that. Okay, so there are some keywords here, guys, of which you should like highlight for you to study smart, because this channel is all about studying together. Let's do revision, and we'll also look at the papers, answer them together if I go live. Yeah, so I'll be helping you guys. So listen, you see the structure. This is what a DNA replication. What is happening here? A DNA is replicating itself. Okay, it's replicating itself like do you see bacterias or viruses? Let's say virus. Virus is when it is found a place in your body, you know, and enter to your cells. It will be affected and it will multiply itself until you get a certain disease. You see, also the DNA structure does what? Replicates. Okay, it makes and an exact copy of itself. That's what I should put it. Okay, so here, okay, it starts where the DNA double strand structure unwinds or unwinds. Okay, as you can see on part 1, you can see that it has changed on how it was before. It unwinds, and the weak hydrogen bonds, they break. Okay, of which leads to what? The DNA structure to unzip. It won't let the nitrogenous bases to connect, so it unzips. And then what the H strand on this, it will serve as a template. Okay, it will serve as a template. So yeah, guys, so those, I think those are the important things you should know. This from the starts from unwinds, and then weak hydrogen bonds break, and then DNA, guys, it unzips. Then comes the template. It's each strand on a DNA structure, because they have separated. Each one will be, well, I mean, will serve as a template. That's how it is. You see, as you can see now, there are strands two strands. Okay, So the free nucleotides build a DNA strand onto each of the original DNA strands by attaching to their complementary nitrogenous bases, of which of what adenine, who it pairs with. It is what they say. It continues pairing with the ones it was pairing with. So nothing will ever change on that. You see. Yeah. So I think guys you heard. If you didn't hear, we can get a simple structure for you to understand on maybe on the next video. So look at this one. Significance of DNA replication. Why, when they say significance, they mean the importance of DNA replication. Why is it important? Okay, doubles the genetic material so it can be shared between the resulting daughter cells during a cell division. I think that's clear enough. Okay. Yes, if you do understand it, that's what matters. English. So if you cram this, you see this. If you do have your phone, just screenshots and make sure you know it. Okay. We're just looking at the time. Okay, it's still early. So here comes on the DNA profiling. So what is it that is used for? Actually, what is DNA profiling? It is a method that is used to identify the relatives, or it is something that is used to find out on some investigations, like someone has been hijacked, we're looking at the fingerprints and all those stuffs, and then they pick victims and all those things. You see that? Yes. So say this DNA profiling, every person except identical twins has her or his own unique DNA profile. It can be described as an arrangement of black bars representing, it's actually lines, it's just a given paper, then they bring up lines, you know I don't know if you know the structure parts. If you all want to see that, I can show you on how it looks like. Okay, so that is how it is used for. Okay, it is used to identify criminals, dead bodies, as I told you, relatives, and paternity, parents, to identify who are your real parents. So yeah guys, I think when I got another time again, I will also post another video based on this, guys. Okay, just relax, study hard, just keep on listening on these videos. Yeah, that's all I can say to you guys. Subscribe to the channel, turn on the notification bell. These is just easy topics, these are just easy topics, but as you can see that I'm using my phone. If I was using maybe a laptop, maybe we would have like written something on this. So I will I think tomorrow or something, I don't know when, but yeah, today we're still focusing on this. I'll also download papers for you guys to understand life sciences better. So let me tell you, on Monday it will be life sciences, Tuesday it's life sciences, Thursday, think, yeah, Thursday, or good, let's say we study in a week, we study a just one day, let's say Monday, Tuesday, Wednesday, Thursday. But you'll see me posting. I don't have to, like, because I do have a timetable here. So guys, stay tuned, turn on the notification bell to be notified every time when I post. Thank you.